you. Okay, welcome to the fourth session of Iranian Insight today, the case discussion and lecture series. And I would like to thank the Sumaira Foundation for the continuous support for these webinars and case discussion lecture series. And today I'm honored to have Professor Waters from Oxford University from Autoimmune Neurology Diagnostic Laboratory at JR Hospital with us. And thank you, dear Patty, for accepting actually our invitation for today, and also for like supporting us with your kind and warm heart every day, like always, for the assay in Tehran. And just a little bit of introduction. Patty is a distinguished immunologist, and I actually myself had a privilege to work with him during this TSF fellowship program, and he actually welcomed one of our colleagues from Tehran University of Medical Science in his lab to help establish the MOGAD aquaporin 4 cell-based assay at Sina Hospital in Tehran. Dear Gila is also today with us. Thank you so much, Gila, for your support, for your effort so far. And Patty, the platform is yours. Thank you so much again for accepting our invitation.
Great. Thank you very much, Sara, Professor Sahraian, Sumaira, Professor Majed, and everybody for inviting me. So this is an area that I'm hugely passionate about, diagnostic assays, as I hope I'll show you in the next 40 minutes or so. It's made a huge impact on patient care in the UK. And from not having heard of these diseases when I started working in the laboratory, we now test 30,000 samples a year to help patients in the UK. So with the help of the colleagues that have just been mentioned, we're hoping to get these live testing set up in Tehran so that we can make an impact on people in Iran as well. So thank you very much for the introduction and for allowing me to present some work here today. Okay, so I've got a pointer here. Great, so these are my disclosures. The main one is that I run a diagnostic laboratory in Oxford where we look for antibodies to different targets in CNS autoimmunity, which is a field that didn't exist 25 years ago. So it's a very new field. So in this field, in the 1960s, we had our first paraneoplastic antibody detection, and that was a HUD protein identified by indirect immunohistochemistry in people with a neuropathy associated with lung cancer. The antibodies that are used to detect these are biomarkers. They're not thought to be pathogenic because the target is inside the cell and the circulating antibodies cannot get into the cell. So they thought they're a biomarker of a T-cell mediated disease associated with a cancer. And often these patients do not respond to immunotherapy. On the other hand, over the last 20, 25 years, started with Professor Van der Len in 2004, 2005, identifying aquaporin 4 as a membrane target for antibodies. So in these surface accessible targets, circulating antibodies can see the protein, therefore they can impact on them. They bind the receptor, they can block the function of the receptor, they could internalize the receptor, which is the same effect. Or in neuromyelitis optica, they fix complement and can cause death of the cell. In NMO, it's an astrocyte. And after that, you get secondary demyelination, and then you get physical signs of blindness, paralysis, bladder and bowel dysfunction in people who have these antibodies. But if they're caught early, the treatments, we have now 3 new verified treatments for these diseases, and steroids work well. Rituximab works well, a CD20 monoclonal antibody. And you can protect a lot of people from severe deficits by early diagnosis and early treatment when we recognize the pathogenesis of the disease. So the first thing that I like to impress is that we believe for detecting these pathogenic antibodies, structure matters. And this is a very nice study by Andrew McKeown and John Mills at the Mayo Clinic, which emphasizes the difference between these paraneoplastic, cytosolic, or nuclear proteins and membrane proteins. So there's an array called HuProt V4. It's got more than 20,000 proteins on this array. Each of the proteins has got a GST fusion attached to the full-length protein, and then it's linked to a nitrocellulose surface with this tag here. Then they use a green-colored secondary antibody against GST to show expression of the protein on the slide. Patient serum is diluted 1 in 1,000 or in the CSF 1 in 50 and run. And if it binds a protein, then this secondary antibody against the human one, Alexis 647, is a red color that tells you that there's an antibody against this protein. So this is what the slide looks like. Each molecule is printed in duplicate. When you've run the tests, so you've had the slide, you put the serum on, you wash, you add the secondary, and then you look for red. The plate is scanned, and the proteins are ordered based on the red signal. What you find with submembrane proteins, so cytosolic or nuclear targets, these are the targets here. And I've just labeled ANA1 is Hu, ANA2 is Re, and ANA3 are Yos. These are classic paraneoplastic targets found in the cytosol of the nucleus. This tells you the serum or the CSF that was tested, and then this is the overall rank of the protein using this chip. And what you can see that many of them are very high up in the ranking. So the brightest signal comes from the targets of the antibodies in the patient's serum or CSF that's associated with the disease. When you go to membrane targets, and many of these are multi-subunit channels or integral membrane proteins, and here we have one that we'll talk about today, aquaporin 4. These are all high positive samples, and the aquaporin 4 serum, aquaporin 4 antibody positive serum, ranked 12,000 on the chip. In the CSF, 18,000. So you can see there's a huge difference in the rank order of these targets when they're expressed in yeast outside of the membrane compared to the rank order of these. That tells me that they need to be sitting in the membrane, and the structure of them outside of the membrane is so poor that the antibodies don't recognize their target. So why do we do antibody testing? Well, here's just a little bit of clinical imaging. This is optic neuritis, longitudinal optic neuritis. This is a longitudinal transverse myelitis. Here's another longitudinal optic neuritis and transverse myelitis. 25 years ago, these two individuals, or four individuals as the case may be, would be considered in the MS spectrum. Other presentations, like signal in the dorsal medulla region or the area postrema, optic chiasma, the bottom end of the optic nerve, and in the transverse myelitis section, there's an H sign. And again, we had no clear idea of what these different imaging features meant. With the identification of these antibodies, these images are taken from the 2015 NMOSD criteria from Wingerchuk et al. So they're all aquaporin 4 antibody positive individuals. We can now correlate these different clinical findings with the disease, with antibody positivity, and compare them with MOG antibody positivity. And we find subtle differences then. For example, there's more anterior optic nerve imaging for the MOG patients. Here you can see optic nerve swelling and the H sign that's identified in many patients with MOGAD. And then you've got more optic chiasmal imaging and more dorsal medullary imaging in NMO spectrum with an aquaporin 4 antibody positive disease. So there are some features that push you towards one or other of the two diagnoses when you have the clinical features, but not all. So the presence of a biomarker is supremely useful. And identifying a target is difficult. So in humans, we've got about 25,000 proteins. If we add splice variants and isoforms, you could end up with over 6 million targets. And in 2024, Van der Lennen, supported by Professor Brian Weinschenker, who looked up the clinical imaging, after Professor Lennon identified patients giving you this staining on indirect immunofluorescence. And this is the first screen used at the Mayo Clinic. And they identified the target in a publication a year later as acapulco-4. And this has had a huge impact on people's outcome of this disease. So this is just a simple survival curve. This data is taken from Brian Weinchecker's data in 1989, and it's 100% of individuals with MS surviving at 5 years. And then Dean Wingerchuk from 1999 showed that within 5 years, 1/3 of individuals with NMOSD had died. With the invention of the assays, the understanding of the pathophysiology of the disease, and the development of more appropriate treatment for these people, this is from a paper last year, and we've seen a 25% increase in the survival of people with acapulco-4 disease over these 25 years. So it's had a huge impact. So what's the target? So this is a topographical map of acapulco-4. This blue is the membrane. So it's an integral membrane protein. And there are 3 extracellular loops from this single peptide. And this is the crystal structure with the loops colored here. There are 2 key methionine residues, methionine 23 and methionine 1. There are 2 splice variants of this protein, and it makes a difference to the structure of the protein in the membrane. So if we take this structure and we turn it 90 degrees towards the screen, we'll see that each of these peptides is part of a tetramer, and it's an obligate tetramer, so it always forms a tetramer. And then you can see where the loops are on this protein. So A loop is central, the C loop is around the outside of that, and then the small E loop on the very edge of the molecule. So this is a target surface for antibodies in NMOSD with acapulco-4 antibodies. And they're found on astrocytes. So if you take a section of brain and you add an antibody here with the gold particle in black, and this antibody is against acapulco-4, this is what you see. So all these particles line up on the surface in pink of the astrocyte on one surface, and you don't see them on the other side. So they're polarized towards the endothelial cells. This is the blood vessel. So this is where the acapulco-4 is present in the astrocyte in the brain. And when we look even closer, these are again gold particles, and these are the acapulco-4, and they form these little arrays. And these arrays are key to the disease. These are the astrocyte end feet. These are cells transfected with the M23 isoform or with the M1 isoform. So those individual tetramers are present when you've got the full-length protein. But when the second isoform is expressed, you form these arrays in cells, and they beautifully mimic the arrays found in the astrocyte end feet in the brain. When we transfect cells with the M1 isoform, so just these dots, so these are cells in the laboratory, we've got a healthy control serum plus fresh serum as a source of complement. And these are 3 acapulco-4 antibody positive sera with another source of fresh serum for complement. Green are living cells, and you might see the odd red dot here. So these are dead cells. With the M1 isoform, with acapulco-4 antibody positive serum and a source of complement, these cells live pretty much okay. Whereas when you use the M23 isoform that has this array structure, now the cells are highly labile to complement mediated cell death. And this mimics the cell death we see in the brain, the optic nerve, and the spinal cord of patients with NMO. And it's the cell death that causes permanent blindness or immobility. So early diagnosis and protection from this complement-mediated death will protect function. So we made assays for the 2 of these isoforms in our lab. So this is a Heck cell. This is the M23 arrayed isoform of acapulco-4, and this is the M1 isoform. When we add, and this is then a cell-based assay, so we add patient serum. If we have target-specific antibody, they will bind the targets, and they bind them in different ways. With the arrayed molecule, antibodies can bind in close proximity to each other. So not only do you have the interaction between the antibody and the target protein, but you may also have FC-FC interaction that supports maintaining these antibodies attached to the target. You may also get divalent binding. So each of these arms are binding a different tetramer on the cell surface, as opposed to when they're at an M1 isoform, only one antibody can bind per tetramer. When you wash the serum and then you add a fluorescent secondary antibody, you can imagine that you get a brighter signal with this form than this. And that's what we found. So these are HEK cells in the laboratory. This is the M1 isoform expressed, EGFP showing co-expression of the M1 isoform. And here we've got the patient's serum in red. So this is a positive test result. But when you transfect the cells with the M23 isoform, it's much brighter. And we can quantify this by flow cytometry. So this is the level of binding here. So higher is more binding. This is the M23 flow cytometry, the M1 flow cytometry. These are relapse samples. It's the same samples here and here. So we can see there's a higher signal when we use the M23 isoform. And importantly, there are some samples that are undetectable on the M1 isoform. And this is very, very important for diagnostics. When we take remission samples, you can see the vast majority of samples on the M23 isoform still say positive. But on the M1 isoform, again, you've got a much group that are negative. And again, the signal is significantly lower. And all of these are from clinically definite NMO patients. So for me, I can see the advantages of the M23 isoform for looking for these antibodies in a diagnostic assay where sensitivity is important. And this is a not to miss diagnosis because you can do something impactful in patients. So I've made an assay. How do I evaluate it or compare it? One of the biggest flaws that we have when you look at the literature for comparing diagnostic tests is you've got one center with one assay and their cohort, and then they tell you how sensitive and specific their assay is. And in a disease where you've got a clinically defined patient group and you've got an antibody to detect in that group, but they're not synonymous, so there are seronegative and seropositive individuals, then this becomes problematic. So in this example here, in black are our clinically defined group, and in green we've got our ACPORN4 antibody positive group. So in the cohort, 80% are antibody positive, 20% are antibody negative. So if you have a test that's 80% sensitive, then it can only pick up 80% of the 80% in the green area. So you end up with a test that's 64% sensitive. There are no antibodies to detect here, so they can't detect them. So that's one caveat of using a clinical criteria to work out the utility of your antibody assay. The second is when you go to another paper and you never know the makeup of the cohorts in these papers. So for example, if this cohort had only 40% antibody positive and 60% antibody negative, and you had a test that was 100% sensitive, it can only pick up these 40%. So it's useful to be mindful of that when you're reading the literature. And the best way and the only way to overcome this is you have to compare antibody tests on the same cohort of patients. And then you compare them head to head. And this is an example of one that we did many years ago now with Sean Pittock at the Mayo Clinic, and Amit Bar-Or, who was at McGill University at the time, and he's now in Pennsylvania. So this was the original indirect immunohistochemistry. We've got a tissue section, put patient's serum on, and you detect with a fluorescent secondary antibody. We compared that with immunoprecipitation, where you extract ACPORN4 from the cell membrane. It's got a fluorescent label here, shown in red. You mix it with patient serum, and then you cross-link and form an immunoprecipitate with a secondary antibody. You spin it down, and then you measure the fluorescence, which gives you an indication of the number of target-specific antibodies in the serum. We've got an ELISA where we've ACPORN4 coated on the ELISA plate. Again, the secondary antibody binds, and you've got an enzyme labeled secondary antibody then, and this gives you colorimetric change, which you can pick up on the ELISA plate. And then we have the cell-based assays that we've talked about just now. So what we did is we agreed with Amit and Sean by telephone on the number of samples. I used old telephones, Sean liked the new telephones, so we'll stick with Sean's phones. So we agreed on the assays that we were going to use, I've just described. And then we did not agree on how many samples, but we did say we use the same tubes and the same volume in these tubes. We coded them, M1 at Mayo, O1 in Oxford to whatever number we were going to use, and everything was done in duplicate. And we sent these samples to Amit at McGill. He added his own samples, which during the study I didn't realize he was going to do. Then he recoded everything and it's sent back to the two centers. So at the Mayo, they did these assays, and in Oxford, we did these assays. Once the assays were complete, all the results and the codes were sent to Amit, who unblinded everything. And then on a phone call, we individually gave in results. And we worked out that we had 60 NMOSD patients and 86 controls in this cohort. And it's a really informative cohort. So just to orientate you, each individual column is a set of results from an individual assay. So flow cytometry or FACS, the live cell-based assay, the commercial fixed cell-based assay, ELISA. There are 2 immunoprecipitation assays and indirect immunofluorescence. Each row is an individual serum sample. Yellow is positive, white is negative. So we talked about the native structure being important. And when we look at the test sensitivities in this cohort, we can see that live cell-based assay by flow or by microscopy gives a higher percentage than a cell-based assay where the target is fixed. And this is better than an assay where the targets are extracted. These assays on this small cohort were all 100% specific, so they're only different on their specificity, and you can see the change in specificities here. And what's nice to see is there's a strong concordance on most of the samples, and then there's a few that pull the tests apart on the test sensitivity. The point I was making earlier, in this cohort, here you've got a bunch of samples. So there's 13 that are antibody negative by any test. So to all intents and purposes, they don't have acapulin-4 antibodies, and they don't tell us about the accuracy of the assay unless there were positives in here. For example, here there's a positive here only in a test that lacks sensitivity. So I do worry that this isn't a clinically relevant test result. But regardless, if we assess these tests just based on the antibody positive cohort, now we can see that this picks up nearly everything, the flow cytometry live cell-based assay. When we go to fixed, we lose about 10%. When we add detergent, we lose another 10% by ELISA and another 10% by PFIPA. So this agrees with John Mill's data that the structure of the target protein is important in these assays. And this type of data you won't pull out if you've got an individual assay on an individual cohort. So what about encephalitides? So just briefly, this is a nice paper by Eric Lancaster, 623 individuals, 731 samples in the clinical laboratory using the fixed kit, and they identified 67 NMDR, 8 GAD65, 4 LGI1s, 2 Caspr2s, and 4 GABA B receptors, a total of 85 positive cases. However, in these tests, you can get indeterminate results, and in this 2-year period, there were more indeterminate results than positive test results. So what Eric did was took all 700 samples into the research lab and used indirect immunohistochemistry. And here's a positive NMDR result. And cell-based assays, these are pictures from my assays. And what he was able to do, of these 97 indeterminates, he was able to resolve 88. 7 had antibodies, 81 were negative. So this is a new 7 that could be reported by using research tests that were not identified on the fixed commercial test. Additionally, the majority of these were negative. And when he screened them, he identified another 10 NMDR, 1 AMPA, 3 LGO, and 1 Caspr2. So 15 new target-specific cases, another 10 that had reactivities on the immunohistochemistry where there was no known target. So these 10 will form a bit of research for Eric. So in summary, with the encephalitides, with the commercial kits, 43 out of 50 serum NMDR samples were positive, which means 1 in 7 cases were missed. In the CSF, it was better, 35 out of 38, or 1 in 12 missed. And then with lower numbers, LGI1, Caspr2, and AMPA in the CSF, 1 in 3 at least were missed on these. So 1/3 of the positive samples were missed. And then for GABA-B and GAD65, they were identified in the serum. So it's just to be mindful in clinical practice when you're using a commercial kit, you get a negative result, you've got to keep your clinical acumen at the forefront of your brain, because it is possible to miss antibody positive cases with the commercial kits that are available. We talk about live versus fixed cell-based assays. So the fixed antibody assay, there's one commercial vendor for this. Live cell-based assays are run predominantly at research institutes, and there's a lot of different live cell-based assays. There's no standardization. So every step that you do in a live test matters. So you can't use the one term for all live tests. But I'll just show you a little bit of the thinking that I went through developing the MOG antibody assay. So what do commercial secondary antibodies bind? So if you go online, you can buy this IgG heavy and light secondary antibody. So in yellow, it binds the light chains of IgG 1, 2, 3, and 4 that are found in your serum at this proportion. And it also binds the heavy chains, so the whole molecule. But this L light chain is common to all classes of antibodies. So it will also pick up D, E, A, or M. So if you have any of these that react with a target on a cell-based assay, this antibody will pick it up. So people are shifting to the IgG Fc. So it only binds the Fc portion of IgG 1, 2, 3, or 4. We never know how good it is binding to the different subclasses, but at least it's sold as binding to all of them. The other thing to note is only 1/3 of this molecule is now recognized by this antibody. So this should give you a dimmer signal than the IgG heavy and light. And as I did with the MOG assay, use IgG1, then there's only a few locations on this one molecule that this secondary antibody will bind to. So it gets you a more specific test, but maybe a little bit lacking in sensitivity. So here's a hex cell depicted with MOG expressed on the surface. These are antibodies in an imaginary patient, and there's IgA, IgM, and the 4 classes of IgG bound to the surface. The first thing you'll note is there's competition for space. So in any study where you're looking for IgM or IgA against a molecule, it might be a good idea to deplete IgG first to relieve the pressure. Then if we look for the IgG heavy and light antibody, on these antibodies, it can bind anywhere on the IgGs, and then on the IgM and the IgAs, just the light chains. So you get a very bright signal. And when you look down a microscope, this is an IgG heavy and light antibody, secondary antibody used against a patient sera that binds to MOG. If you go to FC, well, then you don't recognize A or M. So if they're present, then you lose your signal a bit. And if you move to an IgG1 specific secondary, then it's only IgG1. So the portion of IgG1 on your cell matters to the signal. And this is what we did. So this is our MOG cell-based assay. The patient serum binds the secondary antibody. This is heavy and light, and this is what we saw. So in healthy individuals, we saw a signal half the time when we ran this assay. And these are 3 patients here that we didn't know what they were, but they were all positive. Because we had healthy individuals binding, we decided we'd try changing this secondary antibody to an IgG1. When we did that, healthy individuals, a big bunch of them, became very clearly negative, And then some of these samples became negative as well. And it turns out that these were MS, and these were from non-MS demyelinating patients. So we had a specific test. So we did the same thing, looking to compare. So for MOG, in the literature, it started as Western blot or line blot and ELISA, and then it was moved on to cell-based assays and flow cytometry after Kevin O'Connor in 2007 Nature Medicine paper showed us that the structure of the target in the assay matters. And when Marcus Reindl and I looked at the literature, so this is the literature on immunoblot and ELISA, healthy controls, 14 studies, 10% of 1,700 were antibody positive for MOG when used on a Western blot or an ELISA. In clinically definite MS, 20% of 2,000 were positive, and in non-MS demyelinating disease, again, 20% of 250. However, when we change the assay technique to cell-based assay, then you can see that healthy controls, 45 of 5,500, MS, 26 of 1,500, and ACH4 antibody positive NMOSD, 7 of 1,500, all less than 1% are positive for MOG antibodies by cell-based assay, and the non-MS demyelinating diseases, 30%. So now we have a cohort and we have an assay to detect these individuals, and that's where the field became very interesting. So I'll just run you through a recent paper that we did, although it's not so recent now, in 2020, just looking at ELISAs just to give you a real world example. So red positive, black negative, each row is an individual sample. These are two commercial ELISAs. So these two commercial ELISAs for MOG antibodies do not agree with each other. There's another group of individuals, the same two ELISAs, a different set of sera, and again, the two ELISAs do not agree with each other. These samples were from clear positive, MOG IgG, antibody positive MOGAD patients. These are healthy blood donors. So it's very clear from this data to see that the ELISAs are no use for detecting MOG IgG. In that same study, 7 live cell-based assays were done. Here again are the 39 strong positive samples, and there's a good concordance across the 7 live assays. There is a little bit of discrepancy at the bottom end here. These are the healthy blood donors. They're all black, all completely negative on 7 live cell-based assays. Part of this study included a fixed cell-based assay, and I just want to draw your attention to this. There's a bunch here that are uniquely negative on the fixed test that are positive on 7 live tests. And in 40 healthy controls, we have an irrelevant positive test result on the fixed test as well. And when we look at these samples here in a quantitative manner, so this is the live test, this is the fixed test. So when we look at the live from left to right, if we take this as our strong positive cutoff, then all of these are above this line as positive, and all of these are beneath this line as negative. When we look at the fixed test, so this is our cutoff line, so below this is negative. We now have 4 samples, or 10% of the cohort, that on the live test have titers at 2,500 to 1 in 20,000 that are negative on the fixed test. 10% of them are now on the cutoff of 1 in 10. And again, some of these are very, very strongly positive at 10 and 20,000. And that's the healthy control now positive, borderline positive on the fixed test. So it's just to understand the equipment that you're working with, if you're using live or fixed tests, what metrics to expect, which should help in the clinic. This is another study using the live MOGAD test in Oxford and the fixed commercial test run in the company. These were samples from the Mayo Clinic considered MOGAD spectrum. We identified 25 as positive in Oxford and 23 on the commercial kit. So I take this to be 100% sensitive and this to be 92% sensitive. And when you take the same 2 tests and on a different cohort, so this was done in Oxford and it's the same commercial kit but run in a lab in Canada, you can see there's hugely discordant results, and these results were done for patient care. So it's again just to be aware of the tests that you're using and the possible outcomes you get with them. When we look at head-to-head studies, then these are 3 studies comparing MOG IgG1 positive sera on the live versus a fixed test, you end up with 99% positive, 96 out of 7 are positive on the live test, and 85 out of 97 or 88% positive on the fixed test. And in these 3 studies, there were 374 controls, none of which were positive on the live, and 7 were positive on the fixed test at about 2%. These 7 positives were predominantly MS. So when you're looking for a MOGAD diagnosis and you have individuals who fulfill clinical criteria for MS, it's likely best not to send those samples for testing. And here's a little explanation of why. So if we take in England 5,300 new MS cases in a year and about 270 MOGAD cases, and if we use these samples as our test cohort, and we use the metrics from those 3 published, the average of those 3 published studies, then for the 269 MOGADs, you'll pick out 236 on the fixed test, but you'll also pick up 101 MS cases because they're more prevalent, even though it's 98% specific. And on the live test, you'll miss 3, but pick up 266 of the cases. This means that if you get a positive test result on a fixed cell-based assay, your positive predictive value is at 70%. So it's very easy to see that if you don't send MS patients, then your positive predictive value will improve. And now just a few clinical bits about MOG antibodies. So are they useful? So first of all, MOG titers are not standardized between centers. If I run a test on my IgG1 live assay and Marcus Reindl ran it on his, a 1 in 20 titer in Oxford may be equivalent to 1 in 320 in Austria. So your titers are just going to be based on the individual center that you're testing. When you look at trying to predict a relapsing course from onset titers, this is a pediatric study. The average titer was 1,200 in 72 monophasic children, and the average titer was 1,200 in 44 relapsing children. So the onset endpoint titer of these kids do not predict those who are relapsing or not. And this is work by Brenda Banwell. There were 14 out of 62 children relapsed over a median of 4 years. 38 of those became seronegative and 24 persisted with MOG antibodies. Of the 38, 5 relapsed over the coming years and 9 out of 24 with persistent antibodies. So if you have persistent antibodies in these kids, there's a higher likelihood of relapsing, but it does not rule out relapses in kids who become seronegative. And one other interesting study looking at MOG antibody titers reducing, suggesting a lower risk of relapsing disease in a similar vein. So there were 72 monophasic children, and here are the antibody titers. So the antibody titers dropped by 3 doubling dilutions in the monophasic group, and the relapsing patients, they stayed persistently seropositive with a much lower reduction in antibody titers, even though there was some. The problem is that the follow-up to decide whether you've got decreasing titers or not was 6 to 12 months, and the median time to relapse in these patients is 6 months. So this isn't greatly useful for predicting whether the patient relapse or not. More recently in the literature, you've got IgA and IgM antibodies against MOG. Again, these are 2 relatively small studies. Albert Sayes in Spain identified 2 out of 124 individuals with the MOGAD phenotype who were seronegative for MOG IgG who had MOG IgM, and both were pediatric ADEM. This is the breakdown. So it was 2 out of 35 in the first demyelinating event that were IgM. He didn't find any clinical significance for this MOG IgM discovery. And then Anne-Catherine Probe, still looking at MOG IgA in a group of individuals, found 3 out of 50 with NMOSD that had IgA, but also 5 out of 228 with other demyelinating diseases, and 10 out of 848 with MS. So again, the jury is still out on whether IgA or IgM are clinically useful in these diseases. And the CSF testing, so this is a review of the literature. what we can see is the vast majority, 90%, are antibody positive in the serum. And there's a range of clinical phenotypes in the CSF. Oops. So 1/3 are MS and 2/3 are about the right phenotype. The majority of non-MOGAD phenotypes are in children. So perhaps CSF testing in adults, we think, with the correct clinical phenotype may well be useful, but we need better to understand what's happening in the children's samples. So the current standing for MOG IgG testing, most labs now use 1 in 160 or 1 in 200 as the cutoff. These are healthy individuals. These are NMO, high-risk NMO, ADEM. So these are all within the spectrum of MOGAD. These will be false positive results here. We have clearly negative samples on the test. And then we have this group of intermittents that are not negative, but they don't reach the cutoff. When you're following individuals longitudinally, they may drop into this region. On a diagnostic test, they might be called negative, but they still have circulating antibodies. So it's still not a clean assay compared to acroporin-4. So the take-home messages that I have from this: the native substrate improves detection of pathogenic antibodies. Commercial fixed tests are widely available, easy to use. They're less accurate than live cell-based assays run at certain centers. So it's good to understand the metrics of the commercial tests if you're using them. So it's important to keep your clinical acumen to the fore as false negative results are possible. For MOGAD, limiting testing of clinically definite MS will improve the positive predictive value of your test for MOG IgG. And I'd like to thank the organizers and Sumaira and Michael Devlin for organizing this. And I'm grateful to be invited to talk. Thank you very much.
Thank you so much for the really great presentation. Thank you. And I would like to invite Professor Sahraian as well there for the discussion, opening the discussion session for maybe for 5 to 10 minutes. Do we have any questions, I think? So you can use the chat and also the question and answer icon here, and then I will read your questions that we have. Okay, no questions so far.
Can I start question?
Of course, go ahead, yeah.
Paddy, thank you very much for your excellent presentation, really informative. One of the most important questions at the moment in our country is that we don't have at the moment a fixed cell-based or live cell-based assay. The question of many neurologists is that if we find a patient clinically compatible with MOGAD and we have ELISA test, do you consider to check it in order to confirm in a clinically compatible patient, or it will be misleading? Yeah. Sometimes they check it in 2 laboratories, for example, and if both is positive, they consider it okay, more to go to MOGAD. Or sometimes they check it ELISA twice, for example, after 3 months. As you said, all of them are actually useless. And I wanted to check it with you again because it costs a lot to the patient, as the insurance companies do not actually at the moment accept MOG IgG in some cases. Could you please clarify that? Thank you.
Yeah, so So it's very easy for me. The data on the study we did with Marcus, healthy individuals have antibodies on 2 ELISA kits, and strong positive samples are missed. The 2 techniques, so 2 different commercial ELISAs, did not match up in the vast majority of samples. So the positive predictive value for those 2 tests to definitely identify MOGAD is, it's single figure percentages. It really is not a good way. And remember, these samples that I'm showing you are clear positive. So when you get down to borderline positives, it's going to be even worse. There is 0 value in using ELISA for confirming MOGAD you're much better off to go with the clinical acumen, and you look for features like optic nerve swelling, perineural enhancement, the H sign, and the 2023 MOGAD criteria, the Banwell criteria, is a useful reference.
Thank you. Thank you. My next question actually is regarding the aquaporin IgG testing. And do you think that in future, because in latest diagnostic criteria with the international panel for the MOGAD, for aquaporin testing, is that the best available method? Do you think that in future it will also change to cell-based assay, or it will be kept like this?
So I think the international recommendations at the moment from the 2015 criteria are cell-based assays.
Yeah.
And they're done by flow cytometry at the Mayo Clinic, but there is a commercial fixed kit as well.
Yeah.
So the fixed kit it's pretty good, and it's specific when used in the right hands. You have to validate it at any individual center that's using it, because experience matters. In any study that I've been involved in, when you have the company running the kit, they're exceptionally good at it. So it ends up as a highly specific test, but there are samples that are reproducibly missed on that test, 5%. So it appears that that's a useful test. There are two isoforms that are used. We use the M23, which is a little bit more sensitive than the M1, but it has the caveat that it can pick up antibodies in individuals who don't have the disease. So they're rare findings. So every assay has its issues. So for aquaporin 4, the commercial kit is very useful, but be aware that there's a low percentage that it will miss, but it is generally specific in the right hands.
Thank you. Thank you. Sara, I think we have a question in the chat.
Yeah, we have like one question from Meral from Turkey. So thank you for the presentation. When we had oligoclonal band and also MOG positivity, how should we approach with the test? And when do you think it will be standardized in all over the board? Thank you, Meral.
Great. Oligoclonal bands are found in a low proportion of people with MOGAD. So the presence of oligoclonal bands doesn't rule out the disease. I'm not a clinician. My understanding from talking with clinical colleagues is that sometimes these oligoclonal bands are temporary, so they can change over time, which might be a useful clinical clue. I don't know what proportion of individuals this occurs in. Does that answer the question?
Yeah, I think so. Yeah, of course. Yeah. I have another question, not that much relevant, but anyhow, I think it's really long. Patty, can
All
you
right.
maybe...
There's a question from Brazil.
No, I don't know. No. I think this one is
you?
like... Yeah, it was me.
Yeah.
I wanted to introduce the Weenie to you, but I think he's
Oh my
busy
word.
with the experiment. Yeah.
It's an essay.
Yeah, but the other one is from Solution. Could you maybe read it?
Yeah,
like,
I've had a quick look. I have no experience with this obinutuzumab. Yeah. Yeah.
I think we don't have any more questions. I would like to actually to introduce Vinny, but unfortunately he's busy at the moment with cell-based, and he texted me. But maybe another time. He's also a junior neuroimmunologist from Brazil, and he would like to introduce himself and also talk about a little bit about the cell-based MOG and aquaporin cell-based assay in Latin America, and also specifically in Brazil. And he's now based in Munich, Germany. But yeah, unfortunately, he's busy.
So yeah, I think so. I know Douglas Sato runs live assays in Brazil, so there's at least one center running them.
Yeah. So thank you so much. If we don't have any further questions, I think we can close today's session. And thank you again for joining us, everybody. And thank you, Sumaira, for having us and for put all of us together to have like a really nice and inspiring discussion afterwards. So Sumaira, do you have any?
No, just so glad that we can continue this program and reach doctors and researchers and even patients and caregivers who will benefit from watching this all over the world. Thank you, Patty, for your time and your expertise and dedication to diagnostics. Whenever I think about diagnostics, Patty is the first person I think of. So it's great to have you on here. And Professor, I'm always saying your name wrong, so forgive me. Professor Sahraian. Thank you
You can
so
say
much.
Mohammad, it's easier.
Okay, Professor Mohammad, always nice to see you. And I really, really
you.
respect and admire your leadership in this community, not just in Iran, but I think globally. There's so many people who really look up to you. And of course, Sara for organizing this. And she's a rising star in neuroimmunology, so proactive, so kind, so bright, and really always thinking outside of the box of how can we connect the world by way of neuroimmunology. So thank you to our live audience for being here. This will be available online for you all to reference. Great information here, of course. So just a very heartfelt thank you. And I look forward to seeing whoever will be in Copenhagen for ECTRIMS just in a couple of weeks.
Yeah, thank you so much. Thank you.
Thank you very much. Thank you. Thank you.
Thank you all
Bye-bye.
very much.